All right, so if you remember, one of the characteristics that all living things share is that they're based on the same genetic code, and that's what we're going to begin talking about today, which is DNA and its connection to our traits and such. So DNA, which stands for deoxyribonucleic acid, is a very long polymer. So if you remember what a polymer is any molecule that's made of many smaller molecules. And it's roughly 3 billion nucleic acid monomers long. And if you think back with me, the monomer for nucleic acids are called nucleotides. So in us, in humans, our DNA is roughly 3 billion nucleotides long. That's a pretty big molecule. If you were to be able to take all of the DNA and stretch it out of just one cell, it's about 6 feet long, and it's packed not only inside the cell, but inside the nucleus as well. So this is one little monomer, this is one little nucleotide, and linked several times over with other nucleotides, it creates this, which is, believe it or not, a small section of DNA. This would extend 3 billion bases longer, roughly. But again, our DNA is not one long strand. It's 46 strands, 46 chromosomes. And scattered amongst those 46 chromosomes are about 30,000 genes. When we start talking about genes, that'll make more sense. But the genes ultimately control our traits, and they're scattered amongst and about those 46 chromosomes. And believe it or not, DNA, the importance of DNA wasn't really discovered until relatively recently. When I say recently, I really do mean like the mid 20th century, but prior to that, DNA was, people were aware of it as a molecule, but people didn't quite understand it. And it wasn't until then that people realized the ultimate importance of DNA. So DNA is very complex. What we learned about it is that each nucleotide consists of a deoxyribose, which is this little part here, which is actually a type of sugar. All right. And it contains a phosphate, which is this little part here, and that's the same exact phosphates that are in ATP. That's a common molecule in all of life and all of chemistry, for that matter. And then the very important part is this last part here, which is the base. And between nucleotides, there are 4 bases. Right? No matter what nucleotide, it's always a phosphate. It's always a deoxyribose. But the base is what changes, and there are 4. This one is adenine, which we are welcome from this point on to refer to as A. And another example, notice how the similarity exists between the phosphate and deoxyribose, but that base is different. That is a thymine, or a T. Guanine, all right, looks a little bit like this. Again, deoxyribose and the phosphate are there, but the base looks different. That's a guanine. And then last but not least, you have the nucleotide with the base cytosine. Still the phosphate, still the deoxyribose, but the base again is different. All right, and those bases are really kind of like the letters that spell out your genetic code. A little side note, the adenine and the guanine are both fall under the category called purines. They're larger, ultimately. And then the thymine and the cytosine fall under the category of what are called pyrimidines, all right, which are a smaller molecule. Now, one of the very important people that led us to our ultimate understanding of DNA was a Frenchman by the name of Chargaff. And he found that in any DNA sample, no matter what he took it from, the percentage of guanine always equaled the percentage of cytosine, and the percentage of adenine always equaled the percentage of thymine, no matter what. So here's an example, a little small sample of different living things, very different living things: humans, chickens, grasshoppers, sea urchin, wheat, yeast, and E. coli. So we have bacteria, we have plants, we have various types of animals, you name it. And no matter what, albeit the humans and the chickens are obviously different, but within the human DNA, roughly the same percentage of adenine matches thymine and guanine to cytosine. Look down here to yeast. Roughly it's 27% adenine, 27% thymine, and give or take slightly about 23% guanine, 23% cytosine. Now, the reason why those numbers aren't exact, it's a very tough thing to measure, but no matter, obviously taken with several samples, the percentages always seem to match. A seems to match for some reason to T, G seems to match for some reason to C, and that's the case for all of life. All right, and here's a little example or a little chart that kind of plots human DNA. Notice the adenine and the thymine are about the same, cytosine and guanine are about the same. And we call that Chargaff's rules, meaning that that is a rule that that is the case for all of DNA. All right, and this discovery ultimately inspired, among many others, to inspire Watson and Crick in 1953, a British man and an American. And they eventually won a Nobel Prize for their discovery, and they discovered basically the structure and the shape of DNA, and that it is a double helix, which means it's two strands, all right, and then they are connected and they are wound and twisted, like that's the helix concept. Two strands meaning that they're double. All right, and they're wound and twisted much like that of a ladder. All right. So they furthermore discovered that the backbone, which is kind of like the sides, okay, are alternate between deoxyribose and phosphate, those two parts of the nucleotide, deoxyribose phosphate, and so on and so forth. And then the bases make up the middle. They will connect the two strands. And they're not only connecting, but they're held with weak bonds. All right. And that's important, as we'll see later. The fact that they're weakly held together allows them to separate, and that'll be rather important. All right. But the bases is kind of are the middle, and we'll see the connection in a minute there. And then last but not least, this is where obviously Chargaff's rules come into play, is that for every adenine, it's bonded to a thymine on the other strand. For every guanine, it's a cytosine. For every cytosine, it's a guanine. For every thymine, it's an adenine. All right, you'll never find a thymine bound to a guanine or a cytosine to an adenine or anything thereof. That would be a mistake. All right. So for some reason, adenine matches that of thymine, guanine that of cytosine, and we call that base pairing. The bases connect in the middle that hold the two strands together. Here's a little picture from your book here. Notice the phosphate and the deoxyribose make up that backbone portion. Phosphate, deoxyribose, deoxyribose. And the bases are in the middle. Cytosine to guanine, adenine to thymine, guanine to cytosine, and so on and so forth.